Our first speaker is our guest speaker, Amber Young. She's a second-year graduate student at the Department of Astronomy and Planetary Science at Northern Arizona University. And as part of Dr. Tyler Robinson's research group, she engaged in the search for life in the universe by using computational modeling techniques to simulate planetary atmospheres and atmospheric biosignatures. So specifically, she studies chemical disequilibrium biosignatures and their ability to be remotely detected on Earth-like exoplanet analogs. Amber obtained her master's in physics from Fisk University through the Fisk Vanderbilt Master's to PhD program, and obtained her Bachelor's of Science in Planetary Sciences and Astronomy from Pennsylvania State University. So now I'll turn it over to Amber to talk about the search for life elsewhere.
Hi, everyone. So that was an amazing introduction. I wasn't sure how in-depth it was going to be. So I have a few background slides about myself that I'll just go through a little overview. So again, my name is Amber Young, and I'm originally from Reading, Pennsylvania, born and raised. And I pursued my bachelor's degree at Pennsylvania State University in the Planetary Science and Astronomy department. My junior and senior year, I conducted research with faculty within the Geosciences department, doing mainly climate modeling of early Earth to better understand oxygen and its role in the environment. And upon graduating, I had the amazing opportunity to do a year-long internship at NASA Goddard Space Flight Center in Greenbelt, Maryland, where I continued building on my computational skills. And this time I was putting my focus toward the Martian atmosphere and sort of learning about the role that methane plays within the Martian atmosphere and its astrobiological implications. This sort of helped me transition into graduate school and my graduate journey, where I was accepted into the Master's to PhD Bridge program, which is a collaboration between two universities, Fisk University and Vanderbilt University, both located in Nashville, Tennessee. There, I continued with pursuing methane on Mars as my master's thesis work. And I was a part of Dr. Kevon Stassen's lab, and he was my main Vanderbilt advisor. My second year of my master's degree was pretty hectic in the sense that I defended my master's thesis, and then that summer I got married. And my husband and I made the big move and prepared our cats to move to Arizona, where we both reside now in Flagstaff. And I'm here now starting and in my second year of my PhD program here at Northern Arizona University, working with Dr. Tyler Robinson in the HAB lab. So here at NAU, I've been studying biosignatures and how we may observe those biosignatures in the atmospheres of exoplanets. And in the next part of my talk, I'm going to be discussing chemical disequilibrium biosignatures. And specifically, I'll cover why they're important in terms of the search for life on exoplanets, what a chemical disequilibrium biosignature is, and perhaps most importantly, I'll be discussing its ability to be remotely observed for future exoplanet characterization efforts. So first off, why is chemical disequilibrium important? Well, in the context for the search for life, specifically on exoplanets, chemical disequilibrium has been proposed as a potentially key biosignature for quite some time now. It's a generalized biosignature, meaning that it makes no assumptions on specific metabolic processes at play. And this is important because it leaves the door open for interpreting planetary environments that may be different from Earth. In the astrobiological community, oxygen has been proposed as an important biosignature species to go after in terms of characterization because of its ties to photosynthesis and photosynthetic production here on Earth. But looking for chemical disequilibrium is a slightly different scenario in the sense that we'd be looking for a chemical imbalance within that exoplanet atmosphere. And the only assumption here then is that life produces some sort of waste gas that influences the biosphere in some significant way that leads to the chemical disequilibrium. And this is thought to be a reasonable assumption because, life on our Earth significantly influences the biosphere. In fact, we know that every atmospheric chemical species within Earth's atmosphere is modulated in some way by biology with the exception being the inert species. And so it's thought then that life on other worlds would also significantly impact their biospheres if it's present. So what exactly is chemical disequilibrium? Well, it can be described as a chemical, and I'm kind of using the definition in itself. So let's take a look at an example. So within Earth's atmosphere specifically, we have our oxygen and methane species. And normally those two species would react together to form CO2 and H2O. And that would normally destroy or kill the presence of methane within the atmosphere. But because of biogenic processes like methanogenesis, it serves to pump enough methane or CH4 within the atmosphere to keep its presence maintained at appreciable levels. But how exactly do we quantify chemical disequilibrium? Because we want to be able to observe it, right? So what parameter do we measure within an atmosphere to get at that? Well, we do this by quantifying or calculating a term known as the Gibbs free energy. And basically how this works is, for this example, we're starting with an initial observed state for the atmosphere, which contains oxygen, nitrogen, and methane within the system. And what we do is we calculate the Gibbs free energy, the available energy able to do work within this system. And this is the Gibbs free energy of this initial observed state. Then we allow this initial system to react all the way to equilibrium, such that in this case, the oxygen and the methane form the CO2 and H2O, as we discussed in the previous example. And then you can go ahead and calculate the available energy within this final equilibrium state. And by taking the difference between the two, this is the leftover energy, the available Gibbs free energy that's able to do work within the system. And this is our quantified metric for identifying chemical disequilibrium in a planetary atmosphere. So see the chemical disequilibrium, or the available Gibbs free energy values of Earth, plotted along with other solar system bodies. And you can see that the available Gibbs free energy for Earth is orders of magnitude greater than any of the other bodies shown here. But another result that you can get on the back end of performing this Gibbs free energy calculation is you can solve for the equilibrium abundances, or the concentrations of each species within that atmosphere. And you can compare the initial species, the known observed concentrations, with what you would expect at equilibrium. And so here in this figure, we're comparing the known abundances for every species in Earth's atmosphere, and those are in blue. And then we're comparing those with the equilibrium abundances, which are shown in red. And so you can see here that for oxygen, the initial observed abundance is higher than what you would expect at equilibrium. And this is because of biogenic processes like photosynthesis, which serve to produce more oxygen than what we'd expect at equilibrium. And similarly, for methane, there's way more methane in the expect. And again, that's because of biogenic processes like methanogenesis, which serve to, again, pump more CH4 into the atmosphere at these higher levels. And one thing that I want to point out here is that even though we're using Earth's atmosphere as an example thus far, truly we care about Earth's atmosphere and ocean as an entire system, because the Earth's Gibbs free energy is also sensitive to the chemical species dissolved in the ocean and the reactions that take place between the two. But hopefully by this point, I've convinced you that chemical disequilibrium is an important biosignature and that it can be quantified using thermodynamic equations. But a question that we want to answer is whether or not the Gibbs free energy is remotely observable for exoplanets. And to answer this question, we need to go over a bit of background for retrieving atmospheric spectra. So in a general retrieval process, you start with a data set. You're using a telescope to observe an exoplanet atmosphere, and the data that you get out of it is an observed spectrum. And you have some sort of guess as to what the atmospheric state looks like for that spectrum. So you make guesses about the abundances for the atmospheric species, You make a guess about the pressure profile and the temperature, etc. And then you use a forward model, which in most cases is a radiative transfer model. And you use that forward model to create a high resolution spectrum of that atmospheric state. But because it doesn't have any noise in it, you would then use an instrument simulator to sort of degrade the spectrum such that it's more similar to the observations that you've obtained. And then you can compare that degraded synthetic spectrum to your observed spectrum. And you can use statistical analysis to determine the likelihood that that spectra exists given whatever model you've produced. So a common likelihood or fit metric for this process would be the reduced chi-squared, where a reduced chi-squared value of close to 1 would fit. Chi-squared or the reduced chi-squared value is not near 1, then once you've calculated that fit metric, you can adjust the parameters and then go through the process again and again until you do achieve the good fit. And so ideally, through this statistical process, what you do then is you would compute posterior distributions of the various parameters. The distribution is basically the best fit value to the data, where in this case, what was done was, since we don't actually have direct imaging observations of an Earth-like exoplanet, we have synthetic observations for them, and we have parameters constrained for H2O, O3, and O2 with their posterior distributions. But the moral of the story is that retrieval models help to constrain parameters via their posterior distributions. But another question that we wanted to ask with regard to this is, what is the Gibbs free energy sensitive to? And we wanted to ask this question because, as I alluded to earlier, the Gibbs free energy also depends on oceanic quantities that are unable to be constrained with exoplanet spectral data alone. So here on this plot, what I'm showing you is I'm showing you the Gibbs free energy on the y-axis with the Gibbs free energy for Mars and modern Earth plotted for reference as the horizontal lines. And along the x-axis, we have all the parameters that we've varied for this study. And so we varied the O2, H2O, and O3 according to the best fit +1 sigma and -1 sigma values from the Feng et al. Results, the posterior distributions that I showed a couple slides ago. And you can see that for the oxygen, the Gibbs free energy is quite sensitive to changes with the oxygen, but is not very sensitive to changes with regard to the O3 or the H2O. And for the ocean volume, we basically varied it by an order of magnitude compared to Earth's oceans. And we saw that the ocean volume also, in fact, influences the Gibbs free energy quite a bit. For pressure, we varied it by 10% of Earth's modern day pressure. And you can see that the pressure is not very sensitive, or the Gibbs free energy is not very sensitive to these smaller changes in pressure. And so to conclude here, chemical disequilibrium is a key biosignature and definitely implemented within the search for life elsewhere in terms of characterizing exoplanets with future missions. And we aim to assess the detectability of the Gibbs free energy of Earth-like exoplanet analogs using things like these sensitivity tests that I showed you. And eventually what we want to do is we want to couple the Gibbs free energy models that we have with the retrieval tools such that we can calculate posterior distribution for the Gibbs free energy and see how well we can constrain it for observations in the future. And then in addition to that, as future work, we also want to use different eras of Earth's history as a parameter space for exploring the potential habitable environments that we could find on this planet. So I'd like to give a special thanks to my research advisor, Tyler Robinson, and the NASA Exobiology grant that's responsible for funding this work. And additionally, I'd like to thank my external collaborators, Kat Fang and Joshua Tolton. And of course, I'd love to thank the organizers for inviting me to speak here today. It's been a really wonderful opportunity. And with that, I'll take any questions. Thank you guys so much for listening.
All right, thank you so much. That was a great talk. Does anybody have any questions?
Amber, great talk. I don't work on anything close to this, but I find it fascinating. I was just curious, you mentioned that ocean volume is that there is sensitivity to ocean volume. And I was wondering if there's like why ocean volume as opposed to like ocean area or the fractional ocean area. Is there a way to understand why it's the volume that's important?
Well, that's an interesting question. I'm not sure exactly how to answer that, except that the reason that the ocean volume is important is because it sort of plays into the aqueous phase species that would then contribute to the Gibbs free energy. So the larger ocean you have, the more aqueous species you could have that then sort of enhances the disequilibrium signature in that regard. But that's a really good question. I should probably sort of look into that more to answer it more fully. So I'm sorry.
Well, that's a helpful answer already. Thank you.
All right, Sinclair has a question.
Thanks. Yeah, really great talk, Amber. Similarly to Andrew, I also do not study exoplanets, but it's super cool stuff. Just in terms of trying to detect biosignatures and like the detectability of the Gibbs free energy, besides like it being like a sensitivity issue because you're looking at exoplanets like that are really far away and like having instruments that are sensitive, do you also encounter issues with like the type of star that the exoplanet is orbiting?
Yeah, that's a good point too. So specifically with regard to the synthetic observations that Kat Fang has done, it's specifically geared toward reflected light observations. And so that's because future direct imaging missions like HABEX and LUVOIR are going to be geared toward and sensitive to sun-like stars. And that's great because they're also going to be able to go after the characteristic Earth-like worlds where it's potentially an Earth-sized planet orbiting around a sun-like star, and therefore at characteristic distances for Earth and Sun as well. So it's definitely a lot harder to do direct imaging measurements for different stellar parameters. Stars like M dwarfs and stuff like that are a bit too dim, and it's quite challenging to directly image them because then Earth-like planets will orbit so close to that host star, they're probably going to be within the inner working angles of the coronagraph and for those direct measurements. Certainly there's prospects with regards to doing transit observations instead, where it's more, again, like M dwarfs. But again, for this work, we're sort of looking at sun-like stars because, again, the prospects of having an identical Earth twin out there sort of piques our interest, and that's sort of what we are prioritizing with direct imaging first.
Awesome. Okay, very cool. Thank you.